So, of course, we've talked about variance for quite a while now, but actually there's other types of data that are required for EQTL analyses. So one of them, of course, because EQTLs explain are called expression quantitative trait loci, is we need gene expression data. And gene expression data you've probably seen from the RNA-seq module, but basically the format of it is that you have genes across all the rows. You have usually about 20,000 protein coding genes per cell type, and those constitute all the rows. And then across all the columns, you have the number of individuals in the study. And as I said before, there's hundreds to thousands usually. Sometimes the numbers dip below 100, but it's rare because of power issues of detecting EQTL. So usually you need a minimum of about 100 individuals. And then extremely, because of sequencing costs currently, usually the amount of RNA-seq necessary, the cost becomes prohibitive when you get over, about 10,000 individuals. So the other thing that we need to account for in EQTL analysis is covariates. So very briefly, as I said before, genotypes really don't change over a person's lifetime. However, gene expression varies day to day and by individual and also based on a person's age or their sex. So we do have to account for all these factors in a population cohort. So usually we'll include things like age and sex as covariates in our EQTL analysis, and I'll show a little bit later how we include these. But another very common covariate is age squared, which means that a lot of things as gene expression don't necessarily vary linearly with age. Lastly, we include usually a number of genotype PCs depending on how many individuals are in the population. So 3 is usually a good number, but depending on the population cohort, we can include up to 20.
We'll have a module later about PCA, so you'll know what those are.
Okay. Yeah, we will explain this in a module later about PCA. Yeah. No?